questions today on the biomarker and why can't we study, slow progression versus fast progression, or why don't we have other than ALS FSR score? And that's exactly the problem that we are trying to address and be part of it. This is people's research. We are patient mediated research. So we're doing it because of we belong to the patient group, and we are collecting all this data, and we're inviting the researchers to come in and simultaneously look at the data so we can come to this as fast as we can in warp speed. Somebody put in operations warp speed. That's what we are. So please be part of it. We welcome you to all join hands, because that's the only way we are going to make progress.
Right, folks. It's another gathering of everything ALS. It's your extended family, and we're here bringing Mayo Clinic into your living room today with Dr. Nathan's staff. So each of you, please see the other person on the screen and get to know that they're all in it with you and for you. And so with that, Richard, I'll turn it over to you.
Perfect. And thank you so much, McFinn. And good afternoon, good evening, everybody. My name is Richard Sperry. I'm a new member of the Everything ALS team. It's a real pleasure for me to introduce our speaker tonight, Dr. Nathan Staff. I have a personal connection to Dr. Staff that I'll talk about in a minute. But first, I do want to spend a minute or two going over his very impressive background. So impressive, in fact, that I have a bit of a cheat sheet here that I'm going to have to refer to. So Dr. Staff is a neurologist at the Mayo Clinic in Rochester, Minnesota, and studies disorders that damage the peripheral nervous system. His focus areas include chemotherapy-induced peripheral neuropathy, clinical trials in ALS, and inflammatory mechanisms in post-surgical neuropathies. He received his Doctor of Medicine from Northwestern University, completed his internship and residency at the Mayo Clinic, and was also awarded fellowships in peripheral nerve disorders and clinical neurophysiology, both also at Mayo. Dr. Staff has authored 73 different papers, and among his many awards includes the Mayo Transform the Practice Award for work on refining mesenchymal stem cell therapy in ALS, the topic that he's going to speak to us about tonight. And as I said, Dr. Staff and the team at Mayo, they hold a special place in my heart. In 2019, I visited several neurologists. I received a diagnosis and a confirmatory diagnosis of ALS. In January of last year, I had the opportunity to visit the Mayo Clinic, where I was seen by Dr. Staff and members of the Mayo team. Dr. Staff performed an EMG test on me, and in the results, he and the team detected a conduction block and corrected my diagnosis to multifocal motor neuropathy. For their work and for my change in outlook, I am forever grateful to Dr. Staff and the team at Mayo. So it truly is my pleasure to introduce Dr. Staff and kick off tonight's webinar. Dr. Staff.
Well, thank you. Thank you for the nice introduction. Richard, I wish we could have that sort of outcome for everyone that comes to Mayo. It was, unfortunately, it's a bit of a rare scenario, but it's always so gratifying when it happens for everyone involved, obviously. Thank you so much for everyone at Everything ALS for inviting me to talk today about our work in mesenchymal stem cell therapies for ALS. This group has a special connection for me also. I'd like to dedicate the talk to memory of Peter Cohen. He was a close patient of mine, and when I heard he had passed away, I went to our coffee shop in town and had the tea that he always had when he came to town. And so I was sitting there reminiscing about his passing and enjoying the milk oolong tea that he so enjoyed when he was in town.
Thank you, Dr. Staff. Makes me cry, but thank you so much for doing this. It does, bring great memories, and also we're here with 123 or 120, plus people that we're touching and you're touching. So thanks for what you're doing.
Yeah, it's very much my honor and grateful to be able to be part of all this. We're here to talk about ALS, just to kind of level set for everyone. This is a disorder, as most of you know, of the motor neurons that reside in the brain and the spinal cord, and they degenerate over time, leading to weakness and a number of other symptoms. Looking at the population breakdown for ALS in Olmsted County, Minnesota, which is where Mayo Clinic is located, you can see that the primary group is age 60 to 69, but there's definitely patients on either end of the spectrum age-wise that develop this disease. And much of what we know about the pathophysiology of how ALS occurs is due to the fact that roughly 10% of patients who have ALS have a known genetic cause for that disease. So either their mother or father passed the gene on to them. And the four main genes that cause ALS are SOD1, which is the gene that was originally described and discovered in the 1990s, TDP43, FUS, and then the most common genetic form of ALS, which is the C9orf72 expansion mutation. And so as we've learned more about these genes and learned the different things they do in cells, it's given us a lot of insights into what happens in ALS at a cellular level. So obviously we're all here to find cures for ALS, but there are some challenges around that. As I mentioned, there were four main genes that cause ALS, but there's actually about 30 genes that can cause ALS. So the pathophysiology or the types of ALS is diverse. It's clearly not one singular disease. So that makes it a challenge as to what to target. Another thing to keep in mind is that we've had a tough time taking treatments that we've been successful in animal models of ALS and turning those into good treatments for patients and humans. And that may be due to species differences. So obviously, rats are not people. But also the main animal model that has been used over the years has been this SOD1 mutation model. And it turns out that SOD1 ALS is quite a bit different than a lot of other types of ALS in terms of what it looks like on a pathological slide. And so there's a question as to whether that's a great model to be using to try to treat all kinds of ALS. Another issue is the blood-brain barrier. So there's a barrier that very tightly keeps chemicals in and out of the brain. And so when we deliver pills or infusions, it can be a challenge for those molecules to cross that barrier and get into the brain and spinal cord where they need to act. And then finally, there's always a concern that we're treating people just too late in this disease, and that there may be actually a different type of disease that happens earlier in the disease that might be more treatable than later versions of the disease. And so, we have a need for early recognition. We need to be able to recognize when it's happening, diagnose it quickly, maybe have a blood test that gives us that answer, and be able to treat people at the earliest signs of their disease with hopeful improved outcomes. There are some advantages for doing clinical trials in ALS compared to a lot of other diseases in the same category, such as Alzheimer's. One is the bad part of the disease is that in many patients, it's a fairly rapid progression. And so that actually turns into an advantage when you're trying to study it in clinical trials, because in a clinical trial, you really want to be able to determine a change in a patient over a short period of time. So, in ALS trials, you can see how a patient's progressing and hopefully be able to distinguish a treated versus an untreated group within 6 months, whereas Alzheimer's, it can take years to make that same sort of determination. And then obviously we have a very motivated community in ALS. And so, patients with ALS, families with ALS are very motivated to be part of clinical trials and be part of moving the field forward to cures for this disease. So we're here to talk about stem cells, and I think it's worthwhile to spend just a few minutes talking about different types of stem cells. So the primary cells that are being studied as a therapy for ALS are what are called mesenchymal stem cells, and I'll talk a bit more of those in a bit. But oftentimes when people think of stem cells, they think of, maybe embryonic stem cells, stem cells that come from babies or from fetuses. And certainly that is one type of stem cell. And I like to think of stem cells as kind of the base of a tree. That cell can become every cell in the body when referring to an embryonic stem cell. That cell can become a neuron, it can become muscle, it can become a liver cell. And so, that's one approach to the idea of stem cells. And there's a lot of studies that have been done using adult cells that can be turned into these stem cells that can become any cell in the body. And there's a lot of work in that area. Most of that has been in the realm of better understanding diseases in the laboratory. So kind of lab-based Petri dish studies of stem cells and stem cells that have been turned into neurons. But what we're talking about today is really more the mesenchymal stem cells. And so when thinking about stem cell therapies for ALS, one issue is we like to think of stem cell therapy for ALS as being kind of either neuroregeneration, where the cells are able to make the neurons that are there and sick come alive and regrow, or neuroreplacement, where you put in cells that become neurons and replace the dead or dying neurons. The challenge with that has always been that it's easy enough to get those cells in the right place. You can put them in the spinal cord, you can put them in the brain, but those cells need to send out an axon or a wire and connect to their target. And so, for example, in the low part of the lumbar spine, a motor neuron there needs to regrow an axon and go out to your toes. And so that's a real challenge biologically for the axon to grow out that far and know where to go. It can lose its way along the path. So much of the studies around neuroregeneration or neural replacement are still in the laboratory phase of development. So most of what we think about when we're talking about stem cell therapy for ALS is what's called neuroprotection. So trying to use the cell's biologic properties to keep alive the neurons that are there. And obviously the kind of outgrowth of that thought is that if you treat somebody early enough with a stem cell that's neuroprotective, that they may be left with minimal problems with their neurologic function. And so the type of therapies that have been used, cell therapies that have been used are a number of kinds. So pluripotent stem cells, which is a type of embryonic stem cell. There are neural or glia stem cells. And so the group at Cedars-Sinai with Clive Svensson has been pioneering that area of therapy for ALS. You can imagine a scenario where you're treating with neurons or glia or astroglia themselves, or the mesenchymal stem cell, which again is what we'll talk about more in a bit. And then another issue that you need to think about when talking about stem cell therapies is, are they coming from a patient and going back into that patient? And we call that autologous. Or is it what's coming from another patient? So kind of an off-the-shelf stem cell that can be used for anyone, and that's referred to as allogeneic. And so the issue there sometimes gets to that many of the allogeneic or non-self cell therapies require strong immunosuppressants to shut down the immune system in order to keep those cells around, whereas autologous therapies, or ones that come from the self, generally do not need any sort of immunotherapy. And again, we'll talk about, for our studies, we've been using the mesenchymal stem cells, and they've been autologous preparations. And so the mesenchymal stem cells, so MSC, they reside in a number of places throughout the body. Most regularly, they're harvested either from fat tissue or from bone marrow. And in their normal state, They can differentiate and become connective tissue, they can become cartilage, they can become fat, or they can be bone cells. They can be easily harvested, so that's an advantage to them, and they can be expanded in the laboratory to be used for therapies. And again, it's easy to use these in an autologous format, not requiring any immunosuppression. So what do we think that MSCs do? So this figure kind of cartoons that idea out. One thing that MSCs do and we know about is that they secrete growth factors. And we know from decades and decades of research in the laboratory that there are a number of growth factors that exist that can help keep neurons around longer. So kind of serve that neuroprotective role that I was describing earlier. Some of the growth factors that are particularly implicated are VEGF or GDNF. MSCs also modulate the immune system. Excuse me. So it's thought that the MSCs can kind of change microglia, which are immune cells in the brain, into a more protective characteristic. They also seem to have some impact on T regulatory cells, which are white cells out in the peripheral blood that have been shown to be important in ALS pathogenesis. And MSCs are thought to do some of their actions, at least by secreting little vesicles that include some of the growth factors, but also microRNA. And so digging a little bit more into the idea of the immune system and inflammation's role in ALS, a number of groups have looked into this both in the peripheral blood. And so we've shown that there's altered immune system in the ALS. So this is a study we did a few years back where we took peripheral blood from patients and then basically did all the markers of all of the white cells that exist in the body, and then used a analysis that was unbiased and kind of let the program tell us, well, whose samples are more similar to other samples. And so it's kind of a complicated slide, but if you see all these purple lines up above and then all these green lines below, those are kind of two big groups that split out when looking at the peripheral blood in the group of patients with ALS versus a group of healthy volunteers. And it turns out that the people who were in this green group were much, much more likely to have ALS at about a 75 to 80% likelihood of having ALS versus the purple group, which was about 50-50. And then if you look at those two groups of patients with ALS, whether they're in the purple group or the green group actually lived quite a bit longer. So suggesting that the immune system could significantly play a role in terms of progression of illness and how rapid the disease progresses over time. I think this group has heard from Dr. Appel in Houston, who's done much pioneering work in the world of T regulatory cells, and he's been bringing that into clinical trials. And he's shown that there's a correlation between the amount of T regulatory cells in the blood and how fast a patient progresses. So that's in the peripheral blood, but also there are changes in the immune system in the central nervous system. So here's an example of a PET study. And so this PET, which is a imaging study in the brain, this is the gray shows pictures of the brain and the yellow area shows where it's lighting up. And the particular thing that PET was looking at in this study was the microglia, which, as I mentioned before, are these immune cells that live within the brain. And in patients who have ALS, the microglia activate in the motor areas of the brain, suggesting that they're playing a role in how neurons are being damaged. And then if we look at spinal fluid in patients with ALS, we see that there are increased markers of neuroinflammation. And this just is a graph showing the ALS versus disease mimickers versus disease controls, showing that ALS has more of neuroinflammation than those groups. So I'll come back to this in a bit later. So another question we had as we were developing MSC therapy for ALS was, what happens when these MSCs get put into the spinal fluid. So, like I said, they normally live in the fat or the bone marrow. But what happens when they're put into the spinal fluid, which is a very different environment than they're used to being in? So we did some work in the lab using MSCs from healthy volunteers and showed that, at least on the surface, when you just look at them in the microscope, they look very similar. So there's from 4 different patients, and whether you put it in the normal media, which is the normal broth that we grow them in when they're transplanted into patients, versus putting them in human CSF, they actually look pretty similar. But if you look at how their genes are expressed in those different scenarios, it turns out that, again, the CSF treated, which is this group in the middle, separates out very differently from the normal media. So this is, again, one of these unbiased approaches where we let the computer tell us what groups these samples belong to. And if you do all these different samples and all these different time points, the CSF gene expression data is quite a bit different than either the normal media that we grow them in the laboratory, or if we take out some of the other factors that are normally used in the control media. So suggesting that when these cells go into the spinal fluid, they may look similar, but they're actually changing the genes that they're expressing rather dramatically. And then if we look at some individual genes that we might be interested in, again, comparing the CM to the ACSF, so cerebrospinal fluid, are the main things I want to look at here. The markers of markers of inflammation or anti-inflammation markers are still preserved in the CSF treated MSCs. And we see that growth factors such as VEGF, which I mentioned previously, helps protect neurons, can actually increase expression when they're put into CSF, and we can measure that protein as well. So upshot here is that when MSCs go into CSF, they change dramatically in terms of their gene expression, but if anything, they seem to change in a way that might be favorable toward ALS therapy. So as I was putting this talk together, I was just thinking a bit about kind of how have we taken the idea of using MSCs for ALS, and how has that changed over time? How did that develop? Because I think that many people, when they're thinking about therapies for a given disease, they know that it takes a while to bring it to fruition, but here's how it worked out for us. So in spring of 2007 were the first discussions we had at Mayo about using MSCs for ALS. In fact, it was born out of a group that was studying MSCs, and a person within their laboratory actually developed ALS. And so the idea kind of sprung out of that discussion. About a year and a half later, we started to do the safety studies that were required for us to be able to go to the FDA and talk to them about using this as a therapy. And we were approved to treat a single PALS with MSC therapy in April of 2009. It took us another year to get IRB approval. Again, this was really early on. People didn't know the safety of MSCs at that time, so there were a lot of discussions and a lot of back and forth about using this therapy. But by September, we were able to treat our first patient, who was a nurse at Mayo Clinic. We moved on to a phase 1 study, which is a safety study where we're trying to understand whether MSCs are safe for ALS, and we designed that as a dose escalation trial, and I'll talk about that in more detail soon. And we were planning to enroll 27 PALs into that study. We got approval for that quite some time later, again, now March 2012. And part of that was trying to secure the funding to actually do the trial, which plays a huge role in the ability to make a clinical trial happen. And we were able to treat our first patient in this phase 1 study in August 2012. So we went almost 2 years after the first patient between a single person study into a study of 27 patients. couple years later, as we were moving along with our phase 1 study, we were contacted by Brainstorm, the company that does the Neuron therapy, and we were part of their phase 2 study, along with Mass General, Harvard, and University of Massachusetts, and we enrolled and treated 16 PALs within that group, and finished that within about a year. And all the while we were doing our phase 1 study and finished that in 2015. So that took us roughly 3 years to finish treatment for 27 patients. On the heels of that, we wrote a protocol for a phase 2 study, which is what we're doing now, and that had 60 patients in it. And we're able to, it took, again, almost a year to get our first patient enrolled, primarily due to funding issues. Then we became involved in the phase 3 study, which I'm sure most of you have heard about through Brainstorm. And we treated 39 PALs for that. Once we enrolled our last patient in that study, we restarted our phase 2 study at the beginning of last year, which was sort of slowed down a little bit by COVID, but we were able to move it along. We treated our last patient in Brainstorm in July. And just yesterday, we enrolled our 28th patient in our phase 2 study. So kind of a busy slide, but just to give you a feel of the arc of how these things take time, and that some of that's regulatory issues. A lot of it ends up being funding issues. But we're all working very hard, but things take, are unfortunately slower than we'd all like them to be. So we talk a little bit more about our studies, our phase 1 study. And so what we do at Mayo is we take adipose tissue or fat tissue, and we do a biopsy of that. We bring that to the lab at Mayo, and the lab is able to separate the MSCs from the fat cells. Turns out that the MSCs actually just stick to the plastic that they're living in. So that makes it easy to separate, and the fat floats as does oil. And then in the laboratory, under good manufacturing practices set forth by the FDA, these cells are expanded into billions of cells. And then roughly 2 months after the fat biopsy, they're injected into the spinal fluid via a lumbar puncture, which is a routine test that's done in clinical care. Patients spend a night in the hospital, but have done pretty well. And then again, the idea being that these factors that are secreted by the stem cells may modulate the immune system and secrete growth factors. And so in our phase 1 study, as I mentioned, we had 27 patients treated in that study. Just some demographics about them. The median age was 57. There was a slight male predominance, as we see in ALS, and breakdown of people who had weakness that started in an arm and leg versus speech and swallowing was similar to what you might see in the population. The median functional rating score, which as many of you know is a 48-point scale, where 48 is no impact on activities, versus 0 being totally devastated by the disease. The median number there was 37, but we had quite a few patients that had low FRSs, and that was designed in this phase 1 study. And we had long follow-up with the patients that were in this study, again, by design. And so, as I mentioned, phase 1 studies are safety studies. So they're studies where we're really, kind of first trying it in people and making sure that it's a safe medication to use, make sure that there aren't terrible side effects that we need to be aware of and stop the study. And so to do that, we started with low doses. And so our first 5 patients were enrolled and had 10 million cells injected. So that's 1 times 10 to the 7th. The next 5 patients had 50 million cells injected, and that seemed to be going okay. So we went to 50 million cells with 2 monthly treatments. We ended up treating 7 patients at that dose. And then we went up to 100 million cells with 5 patients, and then 100 million cells with 2 monthly treatments. And we did notice at the 100 million cell level that patients seemed to have temporary back and leg pain. So 8 of the 10 patients in that group had some side effects. So we knew we didn't really want to go higher than 100 million with this cell therapy. But we were also seeing benefits that patients were telling us. So they were noticing increased energy, improvements in their spasticity, limb strength, bulbar function. And so 17 of the 27 patients gave us that report. Now, everybody knew they were getting the injections. So you always have to be cautious about how to interpret an open label study, but it was certainly heartening enough to make us want to proceed with our phase 2 study. This shows the distribution of how the ALS functional rating score changed over time for all of the patients in that study. So each line represents a given patient, and the color tells us what group they were in. And the arrow is when the injections occurred. So you can see, at least in this study, there was still quite a bit of progression in patients after they received injection, but maybe there was some plateauing initially on after the injection. We were seeing biologic effects. And so when we looked at that spinal fluid that was collected at the time of injection, as well as lumbar punctures that were done for safety reasons after the injections, we saw a dose-dependent increase in terms of protein in the spinal fluid cells and a drop in glucose. And when we looked at growth factors over here on the right, we saw that many of the growth factors that we'd want to see change in response to the MSC therapy were changing, in fact, in a dose-dependent fashion. So the top bar shows some of the immune markers, and the bottom panel shows some of the growth factors. Another thing that we were doing in this phase 1 study was look again at the peripheral blood. And you may remember way back in the beginning of the talk where I was saying how different patients had different peripheral blood in ALS, and some did better than others. We saw that when patients were injected with MSCs in this study, that many of the white blood cells in their peripheral blood changed rather dramatically over the course of 1 week. And so we're still looking at this data in quite a bit more detail, and there's nothing major to report at this time, other than that there are definite changes in the peripheral blood immune system when patients are treated with MSCs. So we published that study in 2016, and now we're kind of in the midst of our phase 2 study, which is almost halfway done in terms of enrollment. So as I mentioned in the first phase of the trials, we gave either 1 injection or we did 2 injections spaced a month apart. But for this part of the trial, what we'd like to do is really see, kind of push the limits on things and see what people do if they get the treatments over a year's period. And so this graph below shows how patients come through the trial. The red bar, so we got months along the bottom, so -3, 0 is the first injection. The FB stands for a fat biopsy. So that happens a couple months before the injection, because again, there needs to be time for the cells to grow. And then patients will get an intrathecal or IT MSC 4 times spaced 3 months apart. And then to better understand the cells and do more markers of safety, we have spinal taps in between each injections. And then there's blood draws as well. And each of these arrows up above point to when there are visits for our patients. As I mentioned before, the plan for this trial is to enroll 60 patients. We're almost halfway there as of yesterday. And we're enrolling at all 3 Mayo sites. So while the majority of patients are being enrolled in Minnesota, we do have patients that are being enrolled in Florida and Arizona. The patients in Florida and Arizona do need to come up to Minnesota in order to get their fat biopsies and their treatments, but the in-between visits are all done locally. And I'll say that the enrollment rate that we have is fairly slow. We're enrolling 2 to 3 patients a month. So it's kind of slow going, and that is really based on our ability to manufacture the cells and coordinate the injections.
Your bra, underwire digs, ouch, and back from bra straps is embarrassing.
Hey.
Introducing.
So as always, patient clinical trials have criteria to be in the study. And so the primary inclusion criteria are to have either possible, lab-supported, probable, or definite ALS, be a US citizen or permanent resident, less than 2 years of weakness, and either not on riluzole or on a stable dose for 30 days. An exclusion criteria has a vital capacity, so the breathing function of 65% of predicted, no autoimmune diseases. We're not allowing Radicava in this study, primarily for logistical reasons, and not to be otherwise systemically ill. So the primary endpoint that we're trying to assess for this phase 2 trial is first, obviously, safety. You know, in our phase 1 study, we only did 1 or 2 injections. Now we're proposing to do 4 injections. And in fact, during our data safety monitoring board meeting last fall, we switched from starting at 100 million cells to starting at 50 million cells, because as we're having patients get more and more of these high-level injections, the back pain and leg pain was becoming more of a persistent problem. And so we've adjusted the dosage. So now patients start at 50, but then can increase doses over time if they tolerate it well, which some patients have done a very good job at with tolerating the higher doses as well. And so efficacy is the other endpoint. Obviously, we want to know whether this is beneficial for patients with ALS. And so we're doing that by comparing the slope of the progression compared to a matched group of patients with ALS within the PROACT database, which you may know is a very large database that contains all the kind of clinical data about patients who've been in ALS clinical trials for the last decade or so. And so there's over 10,000 fully de-identified patient records from phase 2 and phase 3 ALS trials. So we're going to compare our patients who are all getting treated in our study to this PROACT database. And then we're also doing a comparison to see how patients do with the slope of the progression in that 3-month lead-in period compared to the time when they're being treated. The other thing we're doing, which I think is particularly important, given kind of what we seem to be seeing in MSC trials for ALS, is that we're doing a responder analysis. So the idea here being that there seem to be some patients that slow down their progression after receiving MSC therapy, whereas other patients don't seem to have an impact of the MSCs on their progression. And so we want to see, is there something different about these groups of patients? If a patient seems to have a good response, what is it about that scenario that's giving them a response to the MSC therapy versus another patient who doesn't seem to have any impact on their progression with MSC therapy? And so, you know, when thinking about it, there's a number of ways that could be happening. You know, one could be the immune system. So we're, you know, again, looking at the immune system quite carefully. You know, I showed that we saw changes in the immune system, both in the spinal fluid and in the peripheral blood in our patients in their phase 1 study. And so we're repeating that extensive peripheral blood immune phenotyping in the patients in this phase 2 trial. We're also collecting spinal fluid and looking at all the various immune markers in the spinal fluid, as well as growth factors and neurofilaments. It could be something about the patient's MSCs. So we certainly know that the MSCs are probably not identical in each patient. So there may be some aspects of the patient's disease course, genetics, that may make one person's MSCs more therapeutic than others. So we're obviously collecting MSCs in all the patients, and we're studying them carefully, just like we did in our healthy volunteer study before, where we're looking at gene expression and as well as secreted factors in the MSCs from patients in the trial. Again, hoping to find a difference between patients who respond versus patients who don't. And then finally, genetics. So we're doing basic genetics of looking at the genes that we know cause ALS, but we also are keeping DNA with the hope that if we see this responder effect, we can do more detailed genetic analyses to find correlations there. And we've been connecting closely with the data scientists and AI researchers at Mayo and getting prepared to be able to do these analyses once we're nearing the end of the clinical trial. So in the last few slides, I want to shift gears a little bit and talk about something that I'm sure is on a lot of people's mind, which is the Brainstorm Neuron therapy. As I mentioned in the timetable slide, we were a site for their phase 2 study, and then we were also a site for the phase 3 study. And as you know, the top line data that was reported by the company and Dr. Sukovich in December at the ALS MND meeting did not meet statistical significance. And they've been in conversation with the FDA regarding paths to go forward. So in many ways, the Neuron MSC therapy is they're similar and different in some ways. So neuron cells come from the bone marrow, and our studies are looking at fat-derived MSCs. And there's still some controversy in the field as to whether those are equivalent types of cells. Some people think that they're significantly different, and other people kind of lump them together as all being MSCs. The other thing that's quite a bit different about what the neuron cells do is that when right before they are injected into patient spinal fluid, they undergo differentiation into a different type of kind of broth that they're grown in that cause them to change their properties a bit. So they're not the straight out of the bone marrow grown up and sent back into the spinal fluid. They change them a bit after they've been out of the body. And that's different than what we're doing, where we're basically taking the fat-derived MSCs, growing them up and putting them right back into the body, and not doing anything special to try to enhance their effects before re-injection. Now, you could argue either way whether it's one's better than the other. I think, ultimately, hopefully we'll be able to get to some answers within the science to determine in patients whether one's better than the other or whether they're equivalent. And I think the jury is definitely still out on that. All right, and so this is the layout of how the phase 3 Neuron clinical trial was designed. Again, kind of looking along here, there were 14 visits. At the green squares at 0, 8, and 16, there were 3 injections of autologous neuron that were injected intrathecally, and then follow up out to the final endpoint, which was week 28. And again, these are autologous cells taken from a patient's bone marrow. And so these are the slides that they shared with me and allowed me to present. These are the same slides that Dr. Sukovich presented at the ALS MND meeting. So this is all public knowledge. And this shows, again, kind of a responder analysis where showing at each week what were the percentage of responders, and that's defined as increasing by 1.25 per month slope on the ALS-FRS from pre versus post treatment. And so you can see at week 2 and all the way out to week 28, there was always more responders in the Neuron group versus the placebo group. Now, as you can see, p-value, this is what we call statistical significance. And typically for scientific studies, we like to see a p-value at 0.05. And so 0.45 means that based on the numbers of patients that were in this group, it did not meet statistical significance. Now, there was always concern that again, there may be some groups that respond more than others, or that the ways that we measure progression in ALS have some flaws associated with them. So they decided before even going into the study that they would look at a separate group of patients who were a little less affected at their baseline. So having a baseline functional rating score of greater than 35 out of 48, and compare that group to those who at baseline were more debilitated by ALS with a functional rating score of less than 35. And it became evident that when you looked at it this way, that a lot of the effect that you were seeing in the combined data was being kind of buoyed up by the patients that had a lower FRS score at baseline. And so if you look at the bottom graph, those with the low FRS, it was very similar between Neuron and placebo. Whereas if you look at the group up above that had the higher FRS, that the Neuron was doing much better. It still didn't meet statistical significance, but it was a much more dramatic change. And one could hypothesize that if you had more patients in this upper group of greater than 35, you might have seen statistical significance. And, I think there's a lot of discussion in ALS clinical trials more broadly about trying to balance the ability to include as many patients in as many clinical trials as possible, basically, opening up the trial criteria very broadly so everybody has a chance to be in clinical trials, which I think is an extremely important goal to attain. But recognizing that what comes with that is that it becomes very hard to differentiate between patients who are responding to an ALS therapy to those that haven't. And when I say that, part of the issue is that there's, as you all know, there's quite a bit of variability in the rates of progression in ALS. And so patients within that population, some have very fast progression, some have very slow progression. And, I think when you look at the data that was put together in the derived study or the Radicava study, they basically designed their study in a way that was going to be the very most likely to find a positive result if it was there. So it was very designed in a very clever way. And it's kind of, basically the patients had to have widespread disease, but it had to be pretty mild. So what that means is that it was widespread, but it was still functional. And so when they were measuring the functional rating score over time, it had room to drop. So, if you have an area of ALS FRS that's scored between 0 and 4, and if you're at 0, you can't go any lower. And so you're not going to be able to measure anything lower than where you're at from the beginning of the trial. So in the Radicava study, they made sure everybody was above 2 in each domain, so they had room to drop. And they had to be patients that were shown to be progressing. So they knew they were going to progress during the time of the clinical trial. And so what they ended up with that was the ability to detect changes very readily in a pretty small group of patients with ALS. Now, it meant that it was a very restricted group. They didn't enroll very many patients and very wide ranging types of patients. So these are the kind of the conflicts I think that we run into. And I think that the Neuron study, at least in the way I've seen the data here, is that they may have fallen into that same challenge where there was too much variability in the groups of patients that were enrolled in the trial. And it made it impossible to detect the difference based on that. This is the last slide from the Brainstorm group, just showing how the ALS FRS changed over time, kind of as you look at each group over the time of the trial. And so the blue is the Neuron and the red is the placebo. And again, the groups that were above 35 showed more change than those below 35. One of the motivating forces for me, the patients that I have currently and the patients I've had in the past, and this is a patient of mine who's was very involved in the wine industry. And after she passed, they had a fundraiser where they made a red wine blend in her honor. And it was referred to as Lucy's middle finger, which is what she was giving to ALS. And so they raised money by selling this bottle of wine. And I have an empty bottle of it in my office. So this is a picture of it taken outside, facing outside my office to look at it every morning.
Thank you for sharing that.
And these are our collaborators, and most of our ALS work has been funded by the Center for Regenerative Medicine at Mayo, but we have received some money from the ALS Association, as well as Target ALS for these studies. And be happy to take any questions.
My name is Sarah. I'm part of the Everything ALS team. It'll be Zoe and myself who will be asking the questions from the chat, as well as questions from patients before that emailed us in. So we'll get right started. First question says, what is the strongest indicator or evidence you've seen so far that makes you hopeful that stem cell therapy can be successful in slowing ALS progression?
It's a great question. I think at this point, all of the patients that I have treated have either been patients I know were treated, so that I didn't know whether they didn't receive placebo in our trials, or they were as part of the phase 3 placebo-controlled Neuron trial. There are definitely patients that I think have slowed progression after receiving therapy. I think it becomes very hard to prove that in a group of patients that you know is getting treated, and because we all know that there's variability in the rates of progression of ALS. But it certainly is enough that makes me want to keep pushing.
So my name's Lily. I'm one of the student ambassadors working with Indu on the speech project right now. And I just am also going to be asking questions. So the next question I have for you is, would receiving one type of stem cell possibly preclude you from receiving others in the future?
Yeah, so every clinical trial has different criteria for being in the trial. Some trials say that if you receive stem cells previously, you can't be in the trial, and that is the case in our ALS phase 2 trial. If you'd received stem cells for ALS previously, you couldn't be in the trial, and that's partly because we need to try to figure out whether the stem cells that we're using are actually beneficial or not. You may find other trials that don't have that restriction in the future, but I think at this point, most would not allow that.
If stem cells are cultivated from an ALS patient's bone marrow or fat cells, is it possible that these new cells retain some of the characteristics of the disease?
Yeah, I think that's something that we need to keep looking at. So, we don't anticipate, and there's no evidence to suggest that these MSCs become neurons. So we don't anticipate that the MSCs would become degenerating neurons just like the other ones from ALS, but could there be genetic factors that come from a patient that make an MSC either more or less likely to be beneficial? So that could play into the issue of some patients responding versus non-responding. So it could be a function of their MSCs, or it could be an entirely different scenario where all MSCs from ALS patients would be less beneficial than if you took an off-the-shelf donor MSC population and treated patients with that. We've been sticking to the autologous or self MSC approach, but there are people looking at the other way as well, and I think that's something that should be explored.
And then how would someone be able to participate in the trial?
Yes, so I realized I should have put up an email. So we have an email that I can put, should I put it in the chat? That'd be helpful.
Yeah, sure. We can also send it out in a post
Okay,
email as
so
well.
it's rstals.research@mayo.edu. And what that does is it goes to a monitored mailbox, and it sends an auto reply immediately that basically asks, to kind of demographic information and tells you about the different trials that are going on and whether you're interested. And then also, if some people use that and end up wanting to come for a clinical visit instead of a research visit.
Hello, Dr. Staff. My name is Zoe. Another question that one of our participants wanted to know. Do you believe it's important for thought to be sporadic cases to have their genomes tested? Why or why not?
So I would say that there's a couple reasons why I would argue to get genetic testing for patients who have ALS. So there's one answer to that, is that there are gene therapies that are coming for the familial forms of ALS. And just because you don't have a family history isn't 100% assuring you that you don't have a familial form of ALS. We've seen data that probably up to 5% of patients who are thought to have sporadic may actually have a C9orf72 mutation, because that particular gene is a little less common to always cause ALS. It can cause dementias, it can cause Parkinson's disease. And so I think that's one argument, that there's gene therapies that are in phase 3 trials for SOD1. There's phase 1 studies for the C9orf trial. They look very promising studies. So if I had ALS, I would want to know for that particular reason, just for my own benefit. It's quite understanding that people may not want to know that information, both on kind of impact for themselves and how it could impact their family members. I would say that if that's the case, that that it would still be great to donate genetic material to one of the large ongoing studies in genetics and ALS, because as I mentioned, there's roughly 30 genes that cause ALS that we know for sure, but there are many, many other genes that likely play some sort of role in whether or not somebody develops ALS. We call kind of risk factor genes. And so the more we understand that, the more we understand about the disease. And that would be something that you wouldn't necessarily need to get the information about if you didn't want it, but it would certainly help, move the research forward.
Kind of changing gears a little bit, can you explain what a data and safety monitoring board does and how it works?
Yes, so it's one of the kind of apparatus that's involved in a clinical trial, especially once you get into phase 2 or phase 3. So it is a group of people that have kind of content knowledge about the trial that you're running, but they aren't at all associated with the trial. So they're totally independent. And what we typically do for these data safety monitoring boards is that they receive all the data and safety that's going on in the trial in the middle of a trial. So the trial's ongoing, and say, twice a year is what we do for our trial. We send all of the safety information and all the data from the trial to a group of people who then review it. Then we talk about it, and then they make recommendations. And that's pretty standard for all phase 2 and phase 3 clinical trials, where you want to have somebody else who's, independent, who isn't, related to the trial, and that may not have, kind of incentive to keep things a certain way. It's a great thing to have. It's great from being the PI in a trial to be able to have a group of experts that you can kind of count on to help out.
Okay, thank you. The next question is, if you believe that Neuron has a clinically significant response that's not seen in the research data.
Yeah, so I mean, it's hard to say. I think when you look at the data that they've presented, I think there is a compelling case to be made that there is an effect there. Now, it didn't meet the primary statistical endpoints that they were hoping for, and that we were all hoping for. I'd love to see the therapy move forward, but I think it's going to, it's hard to predict exactly how that's going to happen.
Can you explain any rationale for having or not allowing Canadians to be in your trial, as well as what it means for slow progressors as to why they sometimes aren't considered in trials?
Yeah, so the Canadian question, that really has been more related to the FDA and our IRB, which is the Institutional Ethics Review Board, requests that we typically have US or naturalized citizens. And part of that is the ability to, the more likelihood that the patients will follow up for all of the visits in a given clinical trial. Admittedly, as I'm up in Minnesota, some from Winnipeg may have a better chance of coming back to visit me than somebody in Texas. They're closer, but that's kind of the hand we're dealt with it. The question about the slow progressors is an important one. So, that inclusion criteria that frequently is in a clinical trial where patients need to have only had symptoms for 2 or maybe 3 years before they can be in the trial, that's kind of getting at that issue of slow progression. And that requirement comes back to this tension between enrolling as many patients with as wide variety of ALS as possible versus designing a study that has the highest likelihood of being able to detect a change during the time of the trial. So if somebody's a slow progressor and say that they're dropping an ALS functional rating score of 1 point every 4 months, if you have a 6-month trial, it's going to be hard to detect any change in that one person. And so, by excluding patients that have had disease for more than 2 or 3 years, that just kind of keeps out that ability to kind of make the power of the study so low that you can't detect changes. It's not ideal, and I think that there are a lot of people that are putting a lot of efforts into clinical trials to be able to prognosticate of how a patient individually is going to progress and use that as the marker for their inclusion in the trial and what they're being based against. And so, there's a number of groups where, you plug in, age and gene and a number of other factors, and it can kind of give you a better ballpark as to whether you're a fast, slow, or mid-sized progressor. And then you can design a trial where you kind of make sure you balance the percentages of low and fast progressors in the same group in each side. So you don't run into a problem where if 2 or 3 real slow progressors end up on one side of the trial, you basically are unable to detect any difference.
Mm-hmm.
Okay, so the next question is that if structured for data collection and conducted properly by PIs in phase 3 trial, do you believe that EAPs could provide additional evidence to help support approval of a therapy?
It's a great thought. That really ends up being a question to the FDA. All data is theoretically data that can be used when looking at whether to approve a medication or not. I don't have enough experience with the FDA to know what data they will use or not use. I guess I'm just going to leave it at that.
Absolutely. Do you have any thoughts on the theory that glycosylation or glucose-treated MSCs can help kind of guide stem cells to maybe damaged or inflammatory sites in the body, and not be so kind of randomly going in whatever direction? And why would that maybe be important?
Yeah, no, that's a great question. I mean, there are a number of areas of research trying to modify MSCs in ways to make them more therapeutic. Neuron is an example of one way to approach that with increasing, growth factor secretion. But I think, all these things are kind of open for debate. We're looking at developing genetic technology and gene therapy within MSCs themselves. There's thoughts of secreting, other sorts of molecules, kind of using the MSCs as Trojan horses. I think this unfortunately, it's a process, and we're still kind of we're not very far down the road yet.
Okay, so the next question is, when do you see a different type of measure for ALS trials, such as biomarkers, instead of an ALS-FRS score?
Yeah, that is huge. It's clear that there are some issues with the ALS functional rating scale, but I will say that at this point that scale has been kind of measured up against everything else that we have out there, and it still performs better than everything else. So that's why it continues to be used. But having said that, there are a number of trials, mostly natural history trials, but also some of the clinical trials where other measurements are being added in to try to find, say, a biomarker, say neurofilament, or any other number of things that are being looked at, and comparing it against the FRS to try to, again, come up with a better measure than the functional rating score. And we continue to try to work toward that. And I think, the ALS community has pushed real hard to continue to make sure that that happens.
I think someone's sharing their eye data with us. In terms of the ALS organizations and patient community you just spoke to, how is it that you see those communities helping to aid maybe trial length or length of time, enrollment time? How can we get more involved in order to help you guys kind of get the answers that we so seek?
Yeah, that is key. I mean, the community, the grassroots and the advocacy within the community has really kept this moving forward. I think it's a multi-pronged approach. There's a need for increased funding at the government level. There's need for increased funding at the foundation level. There's need for keep pushing on the FDA to start thinking about ALS clinical trials in a new way. They've been working on it, but I think I saw a slide recently talking about one of the best medications that's used for brain cancer, for example, thing that's used for everyone who has brain cancer. Nobody thinks bats an eye about it. It's their favorite medication. It's called temozolomide. It prolongs life in brain cancer by 3 months. They don't think about ALS in that way. We all want to have a cure, and that will be an obvious thing when it happens. But I think we need the FDA and the government needs to recognize that if we get enough small wins, that we don't recognize yet whether some of those small therapeutic wins may actually synergize with the other therapeutic approaches that we're having, and can actually start to bring out real meaningful life prolongations in ALS. And so, having a much lower bar for approval of medications and getting it out into the community, I think is really important. And the only group that's going to change that is the FDA. Not sure if I answered all the parts of that question.
You did. Thank you.
So the next question is whether the temporary back and leg pain you mentioned were side effects due to the large number of cells that were injected into the lumbar spinal area, or if it was because of something else.
Yeah, so we think that it is a dose-dependent issue. And I don't want to make too much of it because, for most of the patients that received it, it was more of a nuisance pain than anything. And some patients had zero pain whatsoever. But certainly we were seeing it at the highest dose and weren't seeing it at the lowest dose. And because in the phase 2 study, instead of treating with 1 or 2 injections, we've moved on to 4 injections. We had a few patients where it was really enough that we didn't want to see that anymore. And so we brought it down to the lower dose where we hadn't seen any problems before. And so, yeah, we think it's due to the number of cells. Interestingly, I don't, at least in my memory of treating patients in Brainstorm, we didn't see quite as much of the back and leg pain in the Brainstorm patients as we're seeing in our phase 2 patients. So it's another thing that may suggest they're a bit different.
Okay. I know we're kind of coming up on 7:15, so out of respect for your time, this will be our last question. But whereas the peripheral blood changes that you saw in your study in the immune cells, was that seen kind of further out than just 1 week?
Yeah, so good question. We don't have data on that yet, but we are looking at it carefully. And in the current phase 2 study, the research immune phenotyping blood's being collected at multiple time points. So we'll not only be able to see kind of short-term changes, but long-term changes.
Okay. All right. Well, first and foremost, thank you so much, Dr. Staff, for your time. I know we have a couple of questions in the chat, and if you're open to it, we can email them to you. And if you have time, we would appreciate the answers. But again, thank you so much for your presentation. This was definitely a lot of information to be able to share with our community, and we are really, really appreciative. And I know from our end and how much work you worked with Peter and the rest of our team and the rest of the community for that matter. Thank you so much for that too.
It was great seeing a lot of familiar faces and nice to meet all the people who I haven't met before. And thanks for listening. Appreciate it, the opportunity.
Thank you, Dr. Staff, for making the time, and we probably addressed over 200 people here today. So we are very appreciative of your time, and also brings me memories of 2 years ago meeting you. So thank you for making the time and all your work you're doing to make this a better disease that we can actually, like you said, those little wins, right, to make it into a big win.
Yeah. Thank you so much. Great to see you again.
Thank you. Thank you.
Have a good night, everyone. Stay safe, be strong.
Thank you, doctor. Okay, folks. Thank you, Dr. Nathan. What a wonderful talk we had this evening, folks.
I'd like to invite you all to join our private Facebook group, and nobody outside can see the information that we share within. And we're going to be bringing in a lot more resources for you guys that you don't want to necessarily just post out on social media. So I welcome you all. I put it all in the chat. There's Twitter, Instagram, and then our private Facebook group. So I invite you all to join on that. Thank you.
Yeah. Thank you, Lisa. Thank you, Deb. I just wanted to add, there was a lot of questions today on the biomarker and why can't we study, slow progression versus fast progression, or why don't we have other than ALS FSR score? And that's exactly the problem that we are trying to address and be part of it. This is people's research. We are patient mediated research. So we're doing it because of we belong to the patient group, and we are collecting all this data, and we're inviting the researchers to come in and simultaneously look at the data so we can come to this as fast as we can in warp speed. Somebody put in operations warp speed. That's what we are. So please be part of it. We welcome you to all join hands, because that's the only way we are going to make progress here. So thank you.